Here we have a protein, let's say it's an enzyme, and when this enzyme is activated, it catalyzes a reaction that we can observe, a response in a cell. Now let's introduce a second protein, this other domino, that prevents the activity of the first enzyme, thus preventing a cellular reaction or outcome. The way that geneticists figure out how these two proteins or genes interact is to make individual and then double mutations. So for example, in one mutation, we could get rid of one of the proteins and then observing the effect, and we see that the effect occurs. Look, with a double mutation, nothing happens. In a separate experiment, we take away the other gene that encodes this protein, and we observe that the process, the reaction, the result never occurs. That is, the yo-yo doesn't roll. Here we see the results of the 3 experiments in the presence of either the vertical and the horizontal proteins in experiment 1, where there was no roll of the yo-yo. Experiment 2, the presence of the vertical protein without the horizontal protein resulted in rolling of the yo-yo. And in the third experiment, without the vertical domino or protein, there was no rolling of the yo-yo. The most important pattern that we see in these experiments is highlighted here in yellow, that the effect of no yo-yo rolling corresponds perfectly with the presence of the horizontal protein. This is the result that indicates that that horizontal protein controls the ability of the vertical protein to induce an effect. In comparing both of the experiments where the vertical protein, domino, was present, we see its ability to produce a rolling effect depended entirely on the presence or absence of the horizontal protein. In sum, this protein, the vertical protein, domino, depends entirely on the presence or absence of this protein to produce a cellular effect. That indicates that this protein works upstream of or controls the activity of the downstream vertical protein.